are cooked. This is a representation of a paper from 2021 that studies epiphytic microbiota for foodstuffs, tomato, baby spinach, green olives, and black mission figs. This is interesting for two reasons. Each color represents a type of bacteria. So we see that there is a very different bacterial composition in foodstuffs depending on what you're treating, or they're not all the same. In the case of olives that have been fermented, the main one in purple are bacilli, but there are others that have different composition, and this tells us that we have a great variability depending on the food or the vegetables we consume. Something else that worth highlighting is that on the left we have different microbiomes. The one on these studies is the one before last in green, and this is the diversity index. If we compare it with the soil at the top, the soil has a great diversity of microorganisms, variability of different microorganisms, different components in this matter. In this study, we've also seen a great variability, as you can see from the different colors. The only thing that I would have added to this graph is to show the graph at the same with the same size as the product to know the amount of bacteria ingested, because I could say that the largest amount of bacteria is probably baby spinach. Yesterday in a presentation, Pilar Truchado mentioned baby spinach as well. And there are some vegetable products, baby products, that have a large amount of microbiota that needs to be taken into account. To finish off, I'd like to share my conclusions as to where we have to head towards. This is a research field. And in some years' time, we can use it. And now the way we're using biostimulants to favor the growth of plants and the production, we can use biostimulants to for the plant to contain specific microorganisms and in some way to associate it to properties in health, not only for because of this bacteria, but because of metabolites or chemical compounds being produced, being synthesized. So it's important to consider this, that it's part of the soil. And in the field of production, it's important to observe and see the conditions, the crop conditions that affect this microbiome. How can we achieve that this is preserving the properties that we want in the plant, and what agronomic procedures can be used, because many of them can be affected. The UV light from the sun can affect the viability of this bacteria, and we will have to reach a balance. Also regarding foodstuffs, during the elaboration process or conservation process, we're modulating this bacteria in some way. We are performing some treatments that are affecting the number of bacteria we have and the quality of the latter. So we need to know what is going on to be able to recommend how to use it and what technology to use. And we need to know what's going on in human beings. What interaction is there with those bacteria we have in the digestive system? And what happens with the soil bacteria that are later on found in the human system? Are they alive? Can they perform functions? I'm sure they'll have some modulating role, or they're going to interfere with some mechanisms related to health. In terms of safety, it's also very important. Since we go from the soil to the human being, we have to bear in mind that health is essential and safety as well. So we have to avoid that any dangerous bacteria is out of this cycle that we see in this slide. And the question that I asked about fecal microbiome, what are we going to do with this? Should we discard it? Should we deactivate it? This is a research field of great relevance, in my opinion. And we have to bear in mind that there are two essential tools to study this. One is metagenomics, the study of all these bacteria. There are methods that are very advanced now to study this bacteria with tools that are relatively simple and cheap. They're lowering their costs, and also the metabolomics. We need to know what produces these bacteria and how these metabolites can reach human beings to have an effect on their health. They can be beneficial, but also they can be detrimental. And that's it. This is the end of my presentation. Thank you very much. This is a picture of El Valle de Ricote. Thank you.
Thank you, Francisco. As a comment to your presentation, I have to say that the Princess of Asturias Awards were awarded yesterday, and they were awarded to 3 researchers who were looking for new perspectives on the role of microorganisms to contribute to life on Earth. Now we're moving to Pedro Julián Casas from the company Simborg, who's going to be talking about Globus iranicum. Is the fungi that help us overcome the real challenge of agriculture, being more efficient. These are super fungi. It was created in Murcia, and it is used all around the world. Good morning. I really like the concept of super fungi. I imagine it with a cape. The first thing I wanted to do was to thank the organizers. I am amazed because microbiome is growing and growing nonstop with very good organization. Initiatives such as this one are really important for a sector that is growing more and more, and it's changing the way we see agriculture. 5-10 years ago, this wasn't the case. 5 years ago, things were really different. And thanks to your work and to your trust and to businesses and to initiatives such as microbiome, we're here changing agriculture. So I really wanted to thank you for these initiatives that help us being here today. I don't know if the same has happened to you, but in several conferences I've attended, I've heard a word that is repeated, and it's challenges. Challenges in agriculture. As you know, creating profitable agriculture is more and more difficult, and I'm going to mention some challenges: drought and water stress. Does it ring a bell? This has happened this year. Last week in AgroDigital, I read that a reduction of 30% on the production of cereals is expected this year. I think it's even going to be higher. We also have extreme temperatures, really hot summers, and really warm winters. We are having less and less cold hours for woody crops. Saline soils. This really worries the FAO because plants that need a high amount of water are producing even 50%, are having a 50% lower production. What about production costs? Last year, fertilizers increased their price 300%. It's true that the price has reduced, but things are never going to be as they were in the past. And what about regulatory changes and law changes, farm to fork, reduction in the use of fertilizers, etc.? So this is the current situation. But we have to be positive and we have to see how we can overcome these challenges. And from Simborg, we really believe that we have to be more efficient. I think many of you are familiar with this super fungi, as it was baptized, super fungus, sorry, but I'm sure you're not familiar with all the benefits it implies and with how it can help us overcome these challenges that we're facing and that are more and more usual. There are three benefits: CO2 capture, increasing the capture of CO2, we increase photosynthesis. Let's remember that photosynthesis is CO2 plus water that is transformed into sugar and oxygen. So the higher the amount of CO2, the higher the amount of sugar to produce more biomass. Then we're going to talk about the efficient use of water. And we're going to show that Globus iranicum makes an efficient use of water. And then we're also going to talk about the efficient use of nutrients. Simborg did a trial and we tested our fungus in a hectare to see how much CO2 it could capture. It captured 1.42 tons of CO2 in a hectare, because when I heard these data, I couldn't imagine it. It is equivalent to reducing the emissions of 450 cars in a year. But let's imagine that each hectare of cereal in the world is treated with our fungus. It would be equivalent to reducing the CO2 emissions of 30 cities with the population of New York. I think this is a dream and it can be reached. If we try to think about the agricultural benefits, what does CO2 give to the plant? More photosynthesis, more photoassimilates. But what does the fungus do for the plant to get more CO2? The fungus sends a chemical signal to the plant so that it can capture more CO2. And you will say, Pedro, this is really interesting, but please show it to me. That's what I'm going to do now. This is a scientific paper that was published by the R&D department. This was a crimson table grape with a control plot, treated plot, and in the third one, it was just treated on the first year, not on the second one. As you can see in 2011, this was done with a liquor that if you place it on the leaf and it measures the molecular exchange, it controls how much CO2 the leaf can capture and how much water vapor it can emit. So in 2011 there were differences, and in 2012 too. But please, let's have a look at the darker area. And it means that if we don't treat it, values go back to the beginning. So we increased net photosynthesis in 50%. So each second it increases the CO2 amount by 50%. That's a lot. This is another scientific paper that R&D also published on fine lemon. In this case, just with the control plot and with our fungus. And as you can see, 70 days after application and 150 days after application, these were the results. 20% higher in net photosynthesis activity. Each second, it can capture 20% more CO2, and that is equivalent to more sugar, and therefore more photoassimilates and more biomass, more leaves, more branches, and more fruit. We also say that it can capture more CO2, and that's beneficial. And we say that our fungus is more beneficial regarding the use of water. In 5 tests and 5 papers, we've seen that we can make the most of 25% of the water. But what's water use efficiency? This is not a term that we just made up. This was created or developed by German researchers in 1913. It's more than 100 years old. And what does it mean? It means it is a relationship between net photosynthesis and stomatal conductance. I've got 6 minutes left. Oh my God. Okay, so that's the relationship I was mentioning. CO2 and transpiration. What does it mean? It means that we can generate more biomass per water unit. If we have 2 plant pots with 1 liter of water, we can generate more biomass in the treated plants than in the non-treated plants. And again, these are leaves, branches, and fruits. How is it possible? We can do it in 2 ways. We can make it in 2 ways. This is the same scientific paper. In the first year, there are no statistical differences. In the second year, we can see the differences. And the same for lemon. This is another scientific paper. After 75 days, it's 18%. And after 150 days, it's 25% higher. We also say that we can make the most of each gram of fertilizer. Again, in those 5 tests, those 5 scientific papers that I encourage you to read, we say that we can increase absorption by 30% and with a lower energetic cost. Why? Because we do it in 2 different ways. We increase the root network. If you work with mycorrhizae, you would have seen the difference. It's easy to see because what the fungus do is to modulate the concentration of auxins, and it is sending a signal to the plant. It's saying, if you develop more roots, I will be able to connect with you, and I'm going to be able to send you more nutrients and more water. And what the fungus gives to the plant, you can see the diameter of each hair. That means that nutrients can get to smaller places, and literature says that for each meter of root, a fungus can develop from 7 to 250 meters. So with the volume of the roots, you can see this, that the growth is exponential. And again, Pedro, can you show it to me? I'm going to show you different data. And with glomalin is kind of a natural glue that improves aggregates. It balances macro and micropores so that the roots can breathe and they can retain water. Let's see the first one. Scientific paper developed by the R&D team. This is in melon, treated and non-treated. And they measured the diameter of the roots. Here you can see that there's more volume in the root, a more explored surface on the soil. I'm going to speed up. This is another R&D paper on iceberg lettuce. As you can see, we increased the absorption average in more than 30%. But I was saying we increased photosynthesis. Please have a look at this data. We increase zinc, manganese, and iron. They are essential for the process of photosynthesis. We also increase calcium in more than 46%. Calcium is part of the cell wall and is directly related to firmness. Another scientific paper with lettuce, irrigation water 50% higher, zinc, manganese, and iron. This is the explanation of why we increase photosynthesis, plus all the data in green. Also in lemon, and the same happens. And as I was saying about glomalin, this structural glue that improves the soil structure compared with other fungi. The plant also segregates more glomalin. As a conclusion, we can increase or we can capture more CO2, and that means that we are reducing the CO2 footprint. We increase photosynthesis. We are making making the most of each drop of water that we use and of each drop of fertilizer that we use. I want to thank all of my teams and thank you very much for being here and for listening to me. Thank you.
Thank you very much, Pedro. These new technologies will be sort of forced or implemented by the Farm to Fork initiative that says that we have to reduce the use of fertilizers by 20% at least. Now we're going to introduce Maria Rosa Hermosa, a professor from the University of Salamanca. If we're talking of trichoderma, we always think of Maria Rosa Hermosa because since 1997, that's when she started to write different papers on trichoderma. Conversations of trichoderma with the plant, biostimulation, and indirect biocontrol. Good morning. I'm Rosa Hermosa, and I come from the University of Salamanca. I can see it, but the audience can't. I'm Rosa Hermosa. I'm microbiologist and I come from the University of Salamanca. I've been working with trichoderma since 1992. So I've been working on this for a while in Murcia and elsewhere. Always related with trichoderma. Except in the postdoctoral time that I worked with plants. Yes, the rest of the time I've been involved with trichoderma. Yeah, we have to point out broccoli, but it's been destroyed by now. Okay, here we are. I'd like to start saying that trichoderma, that many of you have talked about, is a fungus, a filamentous fungus. It's asexual in its normal state. It's got over 450 species of strain in trichoderma. They grow in many substrates and they produce lots of spores from conidiophores. In some species, they have been described as having a sexual hypocrea genus, and this only happens in nature, so it's recommended to call them trichoderma, which is the asexual status of genus. The strains of trichoderma are very much marketed, and the largest markets are China, India, Brazil, and we have economic data from 2017 and 2022, and we can see that the markets are keep growing. The strains of species Orsinium, Atroviride, Asperellum, and Viride are the ones that are more quoted as control. Just some information, we published recently a piece of study where we revised the mechanisms where trichoderma carries out biocontrol. And I'm going to mention something regarding parasitism. How can I go back? Parasitism, enzymes and metabolites present activities friends vis-a-vis nematodes and insects. And I'm going to stop a bit in competition and volatiles. In a study from 2011 where we analyzed and compared genomes of three strains from Trichoderma, Perseverance, and Atroviride, both the genome and the transcriptome, when where they overgrew, we could see when creating the phylogenes that microparasitism is an ancestral lifestyle of the genus. All of them have mycoparasitic activities vis-a-vis phytopathogen fungi, and normally there are hydrolytic enzymes in charge of this activity. Trichoderma went evolutionary from parasiting fungi. It moved on to the rhizosphere of plants, and in an evolutionary way, some species managed to become endophytes. Regarding antibiosis, there are many antibiotics described. Trichoderma creates antibiotics against fungi and bacteria with 120 different structures, and are mainly pyrones, pentyl, and terpenes, and glyovirin are the ones, the more relevant ones. In the latest years, this is from a Mexican group, we've seen that enzymes or metabolites created by trichoderma, if they are with vis-a-vis insect pests, we have fall army worms. It is a piece of work with 6 strains, uh, and we can 6 pp and 1 octane, and we see 0.6 milligrams of these metabolites can reduce by 50% of mortality in larvae. I'm going to stop for a bit on this slide. This is a field test that we carried out in season 2018-2019 in collaboration with a ministerial project with ACOR, a cooperative wheat verdant variety with no irrigation conditions in Valladolid. The effects of trichoderma were on the microbiota of the soil, and also the use of nitrogen. Wheat, as you may know, is used for the production. We need it for to produce it high amounts of fertilizers, of chemical basal fertilizers. Here, in the treatments that we compared, well, there was a non-treated control. Then where it says CO2 at the top with two covers of calcium nitrate. Then we had a strain applied to the wheat seed, and the fourth one was a mixture of trichoderma seed and two covers. We took samples in May and we analyzed the microbiota, both from bacterial and fungic microbiota of the different treatments in a metagenomic study. We sampled the root of the plants individually, soil together with rhizosphere and endosphere. It's interesting that the data are clear for fungi and bacteria, that there were or there are three microhabitats that are completely different in microbiota of bacteria and of fungi that are completely separated in soil, rhizosphere and endosphere. Trichoderma modified, as an example, slightly but significantly the levels of some genera of bacteria and fungi, but very different to the calcium nitrate. Both covers caused very big changes in microbiota of fungi and bacteria in soil, rhizosphere, and endosphere. It's interesting that there was an association established between the use of this cover and there was an increase of fungi of different types that are bioindicators of soils that have been abused nitrogen. It was interesting also to see that there was a significant relationship between trichoderma and the increase of some genera of PGP bacteria in the rhizosphere. And curiously, in that moment, we saw that the application on seed of trichoderma was associated with an increase of claridogomus in the rhizosphere of wheat plants. Another mechanism for direct control, we can see that trichoderma plants, this is from a Mexican group and it's quite recent and key, applied to the root of maize plants that could attract parasitoids that control or regulate larvae of insect pests. This is a study carried out where we see that in this rhizosphere of the plant, we quantify the metabolite created by trichoderma, and we can also see that this metabolite or this volatile has to attract the parasite in a correct amount. If it's too low, we don't see an effect, and if it's very high, the effect is not really significant. Here, I'm going to stop a bit more to mention how trichoderma, when it colonizes the rhizosphere of the plant, it reprograms defenses and growth, and its capacity and effect of biostimulation and promoting growth is shown. It increases tolerance of plants to different types of stress, but also abiotic and biotic stress vis-a-vis nematodes, insects, etc. In the plant, we have a priming state in such a way that the plant responds in a more intense way to subsequent abiotic stress or pathogens, and the priming of the inheriting this priming can be seen depending on the trichoderma. I'm going to start with the beginning. The 6pp is from a study group from Naples, where we see the application of 6pp in tomato or colza. We saw how it could benefit the germination of seeds or the growth of plants. I would like to simply highlight that these volatiles have to be in micromolar amounts If you have higher amounts, you don't get a good effect. We also know that many strains of trichoderma, but not all of them, produce auxins as as IAA, indole acid. Like trichoderma, some strains produce, as I was saying, indole acid, and with that mechanism of signaling of auxins in the plant, they can promote growth or development inside roots. I'm going to say once again that auxins have to be in the right amount. Very small amounts don't show this promotion of growth, and if they're in high amount, as you see in the graph in the right, can have negative effects on growth. The optimal amount of IAA would be 10 micromolar. Another mechanism, ACC, deaminase ACC, it has ethylene. This enzyme of trichoderma can reduce the levels of ethylene in the plant, and there is a larger growth. There is a study from Israel from 2010 that silenced in wild strain the gene of ACC deaminase, and they managed to get these transformers in the red box. When they were applied to the plants, the plant grew less. So it was related to the activity of ACC, of aminas of the fungi, with the growth. This effect of trichoderma, as you've mentioned, many of you, depends on the strain, on the plant, on the application mode. Here at the top, on the left, on your left, we see tomato seedlings. We're applying three strains of trichoderma that are different. We could see the positive effects on secondary roots and the growth of the plant, but we cannot see it with all of the strains. Some of them might have even negative effects. This study was carried out in greenhouses with beans, where 20 strains of trichoderma were assessed, and only one showed the promotion of growth. And at the top, we see results of a study carried out with olive trees that were assessed, and when with the sapling, of olive trees, and some of the strains showed growth promotion ability. So it's important to select a strain, and depending on what you want that strain for. Here, it's also important to mention that it was proved that trichoderma promotes growth in a test of tomato in greenhouse. We know that it promotes growth. NPK plus T34, we saw that when we applied salt stress on plants that were treated with trichoderma on the left at the top, it induced as you can see on this slide, when we started to apply nitrogenized fertilizers on the plants, we didn't see that effect. We saw that plants that were treated with trichoderma with NPK fertilization, the defenses of 3 were not applied, but they collapsed, and there was an excess of stimulation. The plant was kind of dizzy because of all those applications. This is a quote from 2021. Pedrero Mendez from Murcia, I have to say. This trial with wheat in greenhouses vis-a-vis water stress. Here we assessed on the left, we see optimal irrigation water. On the left we have, and in the right we have water stress. After 3 weeks, we removed the irrigation for 2.5 weeks, and we have assessed 4 strains that we had removed from the endosphere in the field in Valladolid, even a syncom with a mixture of strains. We observed that not all these strains of trichoderma increase the tolerance to drought. What can we see? That at the top, the plants treated with trichoderma show lower levels of ROS, The mechanism of reactive oxygen species, the with lower water stress, the antioxidant activities increase in the plant. And what we saw when we saw the study at the end for one of the strains and for the Syncon, the sprigs have higher weight. We see that there is a biocontrol activity of trichoderma through the plant. We worked with in a project on olive trees, very sensitive to verticillium, and we saw that plants treated with trichoderma uh, protected them. And what we did here with with confocal microscopy, what we did is that we monitored. This is a confocal microscopy image. I don't know if you can see it properly. We can see trichoderma in green with a line near the vessels that made it difficult for the pathogen to access, that you can see in yellow, to access the vessels in the root. We isolated fewer pathogens when applying trichoderma, and we quantified fewer levels of fluorescence in the root and in the stem of two clones, AC4 and AC15. AC15 is very sensitive to verticillium, and AC4 shows a resistance to verticillium. This is an example of how trichoderma induces priming against nematodes. We worked in a split root system in tomatoes in greenhouses. We got adult plants and we separated the root into different pots, sorry. We applied in one of them atroviride, and in the other one, all the replicas, all the controls, one nematode. After 15 days of applying the nematode, the plants that were treated with trichoderma presented lower number of galls and reduced the number of adult nematodes. If they're physically separated with a split root system, this is a clear demonstration that it is through the plant. Trichoderma induces defenses through the plant. This is my final slide. Regarding a study in collaboration with a group from Costa Rica, working with their strains that were isolated there. We started with in vitro crops, assessing the activity vis-a-vis Sclerotium sepirorum of white rot, one of the strains in greenhouse with the onion. We saw that plants treated with asperellum, this strain showed a lower level of disease. And when we analyzed it from molecular point of view, the plant, we saw that the salicylic root was enhanced. We took the strain to the field and we saw lower level of disease under conditions of tropical climate and higher yield. As a result, is a strain that was selected in a lab, then went to the greenhouse, and then behaved very well or had very good behavior on the field. This is my group of phytopathology and biological control with the newcomers that have recently joined the group. Thank you very much for your attention.
Although most field technicians have included trichodermas as part of their recommendations, there are two reasons why we have to use it. First of all, due to the specifications established by supermarkets regarding the use of phytosanitary products, and the second one is the pillars from the farm to fork strategy in the last 10 years. 2,500 active matters have disappeared. The next speaker is Rocío López from Syngenta, and she's going to talk about microorganisms as a pillar of regenerative agriculture. I think we have to wait. Can you introduce yourself? Okay, that's it. Hi, thank you for being here on this second day of this conference. And what I want to say before I start is that I'm really happy because I can share with you different pieces of information that is directly related to my job and to my personal life. We're talking about that we are currently facing one of the biggest challenges in order to be able to regenerate the planet. We are living a change, a key change, not just regarding climate conditions that are key and that are limiting some factors, and that is setting the trend of businesses. And why? Because each of the people related to agriculture, we are responsible for getting to 2050 and to be able to feed more than 1.5 billion people. And why? Because the key is to increase the production of crops to be able to feed all the existing population. It is clear that we have a problem. But it is not a single problem, but there are different conditions that make us think that everything's changing. We're talking about drought. That's something that we can see that has limited us in the last few months. We're also talking about change and increase, a constant increase in temperatures. We're talking about other factors such as nitrate pollution and the certification of the peninsula. And we are wondering if in the past, we were doing things correctly. So, is agriculture the problem? What we have been working on is in the fact that agriculture is part of the solution to the problem we're facing. We have been talking about sustainable agriculture, agriculture that really needs sustainability as the foundation. But we really need to understand this concept. We have to go a step forward, and we're not only talking about sustainability but about regenerative agriculture. We need to regenerate the planet. We have to regenerate all the resources of the planet. We have to talk about not just about sustainable agriculture as we've been doing, but talking about the concept of regenerative agriculture. And all companies that have participated in this conference, we have to be more aware. And what we want to convey is the importance of looking for solutions, but not just for a single solution. But we have to talk about a 360 degrees concept of crop, of agriculture and cultivation, to find solutions with products that on the one hand are essential, such as phytosanitary products, because they control crop health. We need key products such as the ones we are mentioning today and that were mentioned yesterday that contain microorganisms. We need biostimulants. So what we offer as a company is to get all those solutions, but as a single solution. And in order to do that, we think that as part of that regenerative agriculture, we have to work on a key concept, that's the use of microorganisms in an efficient way. And what does it mean? For some years, we've been listening that the key of the use of microorganisms in the field is to use a strain that can help us doing what we need it to do. We need a strain with specific features for to help us in the key solutions, and we have to differentiate it in a very clear way. As a company, we needed different aspects to treat different aspects, and one of the points is research together with the technical support and R&D department that has been key to be able to say that we are going in the right direction. Here we can see one of our main strains. It's 9690. That's acknowledged at a European level, and this is really important due to its features. I want to highlight different parameters. Tolerance to hard water. We're also talking about everything related to what the bacteria contributes with, such as resistances. We're talking about cysts. This is natural resistance that the bacteria uses to protect itself to fight the conditions that it may face during the crop season. And it also has a PGP activity, that's key. So what did we do? In the last 2 years, we've taken this bacteria and we have developed it to see where we can get to. We created a consortium of microbia with our strain, and we try to find the best partner to potentiate even more those differences we found. And here we found a yeast, and we all know all the benefits that yeast have. This is Saccharomyces anomalous. This is a yeast with a powerful capacity, prebiotic capacity, and it potentiated all the nitrogen fixation, all those features, all the biofilm creation, and in intensive crops, it worked in 25 hours. We needed more solutions. When we are out in the field and we are going to inoculate, when the plant needs it, with an adequate management, we saw that apart from a series of strains, we had to complement it with other strains that did different things. And in this case, we took Thylobacter salinestri because we know that it is essential for the fixation of nitrogen, and we use Bacillus megaterium 9689. And why? Because we have to act in other aspects, in phosphorus solubilization, and we needed a very powerful strain. We also wanted to create a liquid solution, and all liquid formulae out there lost stability in preservation and when used in the field. And with a pool complex, we got it to be stable to have visible results. And what happens? That this is key to say some key data from this strain is that it is endophytous, This is a root sample, how it penetrates the tissue. That nitrogen fixation is key and highly visual. We also have a clear graph where we can see that it not only fixates nitrogen, but it also inoculates and it adapts to the different temperatures. And our strain has got two types of nitrogenases. One of them that is key for high temperatures, and vanadium nitrogenase, that's essential to adapt to low temperatures. And why is this important? Because in the peninsula we've got different soils and different conditions. These are different papers where you can see that what we're saying has been checked and proved. And now I'm going to give you some examples because people ask me, Rocío, at the lab level, from a scientific point of view, this is amazing, but does it really work out in the field? So we did different assessments in the soil where we measured different parameters such as nitrogen fixation, the PGP activity, and the population, the number of populations. And what we found is that in this case we used two different products to see the power that we had. And what we saw is that regarding nitrogen fixation, compared to the control, the capacity increased quite a lot. And when we wanted to assess in the soil the solubilization of phosphorus and all those parameters that are also very important, we did it and we saw that the trend after inoculating, we saw that it went on growing, and that's important. I also wanted to show you results from production. We saw that there were differences, but we had to wait them to see if it was real. This is coriander in the fields of Cartagena. These are pictures, but the weight was heavier. And in the case of celery, the same happened with an increase of up to 18% in production. And this is more money, and that's key for the farmer. Well, we're not only talking about production and about the soil, but when we go to highly demanding crops such as peppers, we see that we have the ability of working with our strains. We can get higher productions with a higher quality. And that's because working with microorganisms is really important to be able to have an homogeneous crop and to get good results. And to sum up, I would like to mention two key points where we see that compatibility between strains. I talked about a solid and a liquid consortium. It's key for them to be able to work in the field. Otherwise, it would be useless. As it happened at the beginning, when people said, I don't use microorganisms because they don't work. So what we did was to do many tests and compare them with different products in different conditions to verify that apart to what we're saying, apart from the features of strains, apart from the soil analysis, etc., they have to be compatible with products that the farmer uses on a daily basis. And we got successful results. Rocío, 2 minutes. Okay, and in the end, what we see is that We are taking part of a regenerative agriculture, and we are helping the soil. Without further ado, these are many different results that you can check, and we can share them with you. And at the end, looking for strains that really work, it's really important, not just at a European level, but also at a national and international level. Thank you very much.
Thanks to the use of these technologies, we'll be better technicians because we'll save costs, time, and we'll increase our production or our yield. I don't know whether we'll be able to feed all this population, but anyway. Our next speaker is Manuel Valenciano from Alga Energy, and he's going to talk about microalgae and in their relevance for the health of soils and yield of crops. Manuel, you have the floor. Good morning, everybody. I'd like to thank the organizers. It's a very important event for us. We have to generate spaces where we can exchange opinions and transfer knowledge, both at an academic level and at scientific level, private companies, farmers, technicians, a gathering of all of us. It's interesting. The only thing, agro marketing, please don't hold dinners the previous day dinner because we come here in bad shape, but it's very interesting, I have to say. Throughout the sessions yesterday, I talked with some of you were critic because some of the presentations were kind of commercial, but it's important that we share all this among all of us. And behind commercial presentations, there is a great effort of development, of research, economic investment, where business people have invested money and time to try to improve this world. So I'd like to acknowledge their work. The organizers have done such a good job that, as Francisco was saying before, yesterday the Princess of Asturias Prize was awarded to a group of researchers that have considered as key the use of microorganisms for life. It's something like that. I can't quote literally, but it's really timely. It comes without further ado. I'm going to try to stick to my time so that I'm not told off. I'll try my best. I'm going to talk about microalgae. I want to share with you this great world of microalgae. They are the origin of all plant life and responsible of oxygenic photosynthesis. Many years ago, there were some changes in the atmosphere, and aerobic respiration was created, and algae were to blame for this. So we want to highlight microalgae and their role. Microalgae in the soil capture about 4 gigotons of CO2, but it has the capacity to capture 200. And this is carried out by this makes it an extremely important element. They have the photosynthetic ability 10 times more than land plants, and this is something we want to highlight. They are responsible for 50%. Is it me? 50% of the oxygen that we breathe. It's a microorganism or a microscopic organism, as I was telling a colleague, that it's essential for life. And microalgal photosynthesis is an important component not only for aquatic or marine ecosystems, but also for land ecosystems. It is fundamental for land ecosystems, and it's everywhere. It even has the ability to adapt for microalgae to be found in extreme places in the planet, that you can find microalgae there as well. To preserve the diversity of the soil, well, this is our responsibility. It's never been as important as it is today to reduce CO2 emissions and manage to keep our life being important. Microalgae have a very special, very particular composition. They have over 50% of their composition is proteins. This makes it very special, different. When we talk more colloquial or microalgae or micro or marine algae, this wealth is different, but they not only have they have this high content of proteins, also lipids, also carbohydrates, and another series of compounds that are very important for a practical application in agriculture, like vitamins, minerals, carbohydrates, pigments, chlorophylls, for a practical application in agriculture. What we've developed is that starting with this raw material of microalgae that is unique and that cannot be achieved anywhere else or any in a different way, universal, we've obtained a process where we can extract 100% of the bioavailable elements in their composition and generate a biomass that subsequently will be subject to technology that we have patented and registered, UPT technology, where we can make sure that is stable and maintained, and then the composition is improved with some elements that are very important, such as peptides of high molecular weight. In this, the Institute of Biochemistry and Bio and Photosynthesis from the University of Seville, Professor Francisco Romero, that we'll talk later, has participated and developed important trials together with Dr. Miguel Garcia Guerrero, that is our chair and scientific director that has been researching 50 years in the microalgae world. And we see the application of microalgae creates critical genes or genera for crops, creating transcriptomic movements. I'm not going to talk about this topic because I'm not an expert, but it will be explained. We generate this kind of response. What we did was to put in practice trials everywhere. We've carried out all sorts of trials, and we keep saying that we're having an increase and an improvement in the yield of all crops in very different conditions. And also we've subjected this these trials to the use of fertilizer, reduction of the use of fertilizers. And in all cases, or in most cases, we've seen that we were causing an improvement in results, and we wanted to analyze everything because and now we're going to talk about this, but the truth is that we need to explain the action mode of our actions and why these changes are brought about. We're not going to forget that we're going to talk about stimulant to be applied on the soil, and biostimulants are defined by the new European regulation as to what they do and not by what they are. So we know what they do and we want to know why they do it. From then on, we are going to present in this conference a job carried out by the team in Sebasisik in a plot near here. So we're talking about a territory that we know in Santomera, where the team led by Carlos Garcia and everybody in Sebas has made a great job, and they wanted to study what happens when you apply biostimulant and why microalgae are so interesting. So we analyze different parameters in the crop of young and adult almond trees, mandarin, apricot, and lettuce. And we're going to share some results. To see what was happening in the soil, we have to know what happens at carbon level, organic carbon, and the content of humic substances. As we know, we talked about carbon yesterday. There was a presentation on carbon. It is a very valid indicator of the health of soils, and with the application of our biostimulant, based on microalgae, we could increase the content of carbon in soils after second and third application. This was happening, and this might happen. This also happened with humic substances. That is the more stable source of carbon that we can find in the soil. So we could see that something was going on, that something was happening, and in some way we were influencing positively in the carbon cycle. We'll get into detail later. We did the same thing with nitrogen to see what kind of effect had our application of biostimulant on the soil in the nitrogen cycle, total nitrogen and hydrosoluble nitrogen. And the same way we saw it before, we've seen that with the application of biostimulant, we increased considerably the contents and fixation of nitrogen in the soil, both total in the case of nitrogen, total nitrogen, in the case of nitrate or ammonium. So with the application of our biostimulant, we saw that we were causing a positive effect on the nitrogen cycle. And we went to do it with phosphorus and potassium as well. In the case of phosphorus, the results were not as clear. We saw some kind of trend. And in the case of potassium, they were not conclusive because the results were not very clear. In the last application, the content of potassium was higher, but these were not conclusive results. So we want to see what happened with the basal respiration, and we saw that expressing it, the team from Sebas said that when there were high increases regarding basal respiration in the group, both in the case of young almond tree and adult almond tree, and the results replicated in other crops. I don't want to get into detail now. And this increase regarding the activity of basal respiration implied favorable response in the biogeochemical cycles that we mentioned yesterday, and this caused a higher assimilation of nutrients by the crops. We analyzed why this happened, and the team said that, well, some of the enzymes had to do with this, and it was analyzed what happened with the dehydrogenase enzyme, which is an indicator that is similar and works in a similar way to basal respiration. And we managed to see that there was it was positive. There was a positive effect on this enzyme on the soil. Here we mentioned in some presentations yesterday. We talked there were presentation about B-glucosidase activity. So I wanted to see what happens when we apply this biostimulant. What effect is causing this enzyme that is positive for the development of crops? We analyzed beta-glucosidase activity on the carbon cycle, and it was responsible for hydrolysis in some celluloids and in the termination of beta-glucosidase, and the effect was very positive. We analyzed what happened with phosphatase as well. Yesterday we heard about phosphatase as well, and there was a higher activity of the phosphatase enzyme when applying our biostimulant. The same happened with urease and glycine aminopeptidase. All these enzymes that were involved in the carbon cycles, in the phosphorus cycle, in the nitrogen cycle. And as we can see from the very beginning, there is a higher activity in the development of this kind of cycles. This is important and makes us think about it. If we manage to reach overexpression or improvement regarding assimilation of some enzymes, we'll probably have to adjust, and that's what we're working on, the doses of some of our fertilizers, because this can imply a reduction in the use of some nitrogenated fertilizers. In the case of glycine aminopeptidase, and they are indicated for the hydrolysis of some proteins, and we were contributing the development of specific amino acids. So in all the cases, we saw a favorable evolution and a positive response by the enzymes when we when we applied our biostimulants based on microalgae. The team wanted to study the structural diversity and the functional diversity because it's important to know what is going on. And we were surprised with the data because the increase is considerable in the soil regarding the development of some fatty acids from phospholipids, sorry, they were more positive than negative, and the appearance was compared. The values are low, but the reality is that we started with bacteria content that was very very low or even near zero. But we saw the appearance of actinobacteria and a strong increase regarding some elements, not only at structural diversity level, but also it worked at functional level as well. And there was an increase in the consumption of some bacteria that could have a positive influence. So to conclude or recap, I would like to say that with the application of our biostimulant based on microalgae, not only did we achieve an improvement in the assimilation of some nutrients, but also an improvement regarding the structural and functional diversity of what we found in the soil. All this information, all these trials have been verified by official bodies, also at private level, and we saw the same results. So to conclude and to stick to my time, um, these 15 minutes that were given by the organizers, well, according to FAO, 180 hectares of soil that can be cultivated has been degraded and has been lost. We have to raise awareness, and we have to know the importance of soils and how quickly we're losing them. Microalgae are a very strong biostimulant and play an important role to preserve soil and to improve biodiversity. Microalgae produce critical genera that can be seen even 2 hours after the application of our biostimulant, and they are maintained over time. And also, they intervene in a very favorable way in the cycle of different nutrients, influencing very positively the availability of the latter in the plants. I wanted to explain what microalgae are. They're a universe, a great world that I'm happy to share with everybody. As a continuation of all this research group, we created, no, for this research work, we created a group with Sevasic and other entities and organizations like Asaha. And we created an operational group, Microclimate, where we kept assessing things, and I invite you to visit and see the results, and maybe the next paper will mention something related to this. Thank you very much, and congratulations for the organization and wonderful food.
As the Minister Luis Planas said when he visited Alga Energy Company, microalgae are going to have a key function to develop a more sustainable and profitable agriculture in the future. And now Francisco Romero from the University of Seville is also going to talk about microalgae as biostimulants. Good morning. First of all, I want to thank the organizers for inviting me. I'm Fran Romero. I'm a professor at the Computation and Artificial Intelligence Department at the Institute of Vegetable Biochemistry and Photosynthesis. It's a collaboration between the University of Seville and the CSIC. I don't know if you can see my presentation. Today I'm going to talk about some results that we had as part of the microclimate project that Manuel mentioned to improve resistance of crops to drought by using microalgae. most part of my presentation, I'm going to talk about wheat, tomato, and Arabidopsis thaliana, but I wanted to highlight that the vegetable kingdom has a wide diversity regarding physiology and morphology, from crops that are essential for humans, such as maize or corn and wheat, to non-vascular plants, such as mosses, getting to organisms such as Klebsiella or Streptococcus. All these organisms can do photosynthesis. They have chloroplasts that get the sun rays and they channel it to break the water molecule. This releases oxygen and it allows living other organisms in the world, and they use energy from this process to fixate CO2 as glucose, and these constitute the basis of all trophic chains in the earth. These organisms are highly attractive to generate applications that are sustainable because their development is related to the capture of CO2 and to the accumulation of CO2 deriving from the climate change that we're suffering. The vegetable kingdom has divided into two main groups: plants and microalgae. But with the recent development of omic techniques with genomics, we have found the genome of all these plants, and we've seen that what we thought that was very different is not that different. There are groups of microalgae that are different from plants, but there's evidence from these phylogenomic groups that there was a single event during evolution that created real plants, Viridiplantae, that include two groups of microalgae, chlorophytes and streptophytes. They share molecular features with common plants. Response to biotic and abiotic stress, these are features that are shared by these two groups. So it is not surprising to see that microalgae extracts have a biostimulant effect on plants. However, molecular aspects were unknown, and this was the beginning of the microclima project that Manuel mentioned. This is a collaboration, public and private collaboration with different institutions with specific roles. Alga energy generated two biostimulants based on microalgae, LRM and Bio2. Thebas, as Manuel mentioned, analyzed the effect it had on the soil of these biostimulants, and together with Imidra, we did a phenological analysis of how plants developed once they were treated, and I applied omic techniques, transcriptomic techniques in plants treated with biostimulants, subject to drought and non-subject to drought. BioVegen was in charge of the website that I encourage you to visit to have more information. Within our tasks, we had two main blocks. One of them, the characterization of LRM and how it acted on wheat and tomato. The other block wanted to apply our transcriptomic techniques in the development of new biostimulants, Bio2, that focused on microalgae and different types of processing those biostimulants. So, starting from wheat, we had plants with drought combined with foliar spray treatment of biostimulant. We extracted RNA and applying different techniques, we estimated the expression levels, in this case, of those plants subject to drought, treated and non-treated. Each dot is a gene that is activated or suppressed. If it is activated, it's red, and if it is suppressed, it's blue. So what we saw was that the foliar application has an effect on the gene expression on wheat over 1,000 genes. We applied AI techniques regarding the information we had about these genes, and we saw that they were not randomly distributed in the plant functions, but they focus on the response to water deprivation. That is, plant treated with biostimulant has a higher response to water deprivation. We also saw the processes that contribute to accelerating the plant development. This led to the assimilation of phosphates. Some examples to show you that gene induction is not pure, but that the changes depend from different factors, and the genes that regulated that response to the lack of water in the case of wheat refer to the control of stomatal closure. At a transcriptomic level, apart from that analysis, we validated it monitoring how plants developed. These are plants subject to drought, one treated with sprayed water, and another one, the one treated with the microalgae extract. In the first stage, we saw that plant treated had more branches. In the second stage, we saw that it was taller, that the treated plant was taller, and both plants developed a single shoot. But the plant, as I was saying, was taller. What we see in that case, it was also seen in plants not subject to drought. Here, even in the first stage, we saw that non-treated plants in treated plants there were more branches. In the second stage, it was taller too. And in the final stage, the plant was more dense. We validated it getting different elements, and we saw that there was an increase of more than 4 in the case of sprigs and in the grain weight. These results were not as spectacular in the field, but Asaja had similar results. What we saw in plants with drought was also seen in plants not subject to stress. In this case, the production was higher, but we saw an increase in the production in plants treated with the biostimulant. So what we wanted to see was if this biostimulant was also only effective in one crop or in different vegetable species. tomato is very different from wheat, so that's why we chose it. We also did our transcriptomic research. Again, these are plants subject to drought, and we saw that there were thousands of genes that were not randomly distributed, but they were focused in response to heat and in development processes. In this case, the genes that were activated were slightly different to the ones in wheat, but it's because they're different plants and they have different strategies to face drought. Wheat closes stones, but in tomato, it accelerates the flower transition. In order to prove what we saw in the lab, we did a monitoring of plants. Here we can see that plant treated with LRM shows a lower rate of stress. The same happens in plants not subject to drought. And I wanted to show you a video. I don't know if they can play it, because in the case of tomato, we use ubiquitous computing, and we placed a camera to see what the response was after a period of complete drought. On the left, you see the plant treated with the biostimulant. You have to point at the broccoli. The second part was to help the company developing new biostimulants or new formulae to stabilize biostimulants they were working with. As we were working with a new biostimulant, we used Arabidopsis plant with the same foliar treatment. We did the sequencing, and this biostimulant that made the most of microalgae, we saw that it altered gene expression traits, and it focused on cellular response to stimuli. We were able to compare genes which activated in a biostimulant and the other one, and we saw that they share some genes, but they have different modes of action. Finally, as we generated a new product, we had to analyze the new preservation ways and the stabilization, etc. So we received different biostimulants, and at the lab, using our cultivation systems, we analyzed the use of 4 different microalgae to see if they had a biostimulant effect or not. So applying bioclustering techniques, we compared the treatment of these biostimulants with treatments with water spraying. Using AA techniques, we were able to identify 3 different groups. On the one hand, we saw that there were some groups from products that were paired with water, and those products didn't have any biostimulant properties, and they activated a reduced amount of genes. We also identified another group of biostimulants that were generated with different processes, enzymatic processes and different parameters, and there we found 2 microalgae we had used, and these new products had a low and medium effect regarding the amount of genes affected. And finally, we found stabilization processes and a new microalgae because it had a massive effect on stabilization of the gene expression on plants. This was carried out on an Arabidopsis thaliana. the next step would be to test it in other crops such as wheat and tomato. I would like to finish off by saying that there's a huge diversity in the vegetable kingdom, and as part of it, there are two groups of microalgae that are very similar to plants. They share a lot of molecular systems, and therefore their extracts are very good to be used as biostimulants. We have analyzed two biostimulants, the LRM, that has a clear effect on resistance to drought on wheat and tomato, but in wheat it induces closing the stones, and in tomato it accelerates the flowering time. And that new biostimulant has a biostimulant power, that bio2, and we establish a protocol to be able to validate new biostimulants and to classify them according to their low, medium, or high power. Of course, this has been carried out by a group. I want to thank Mercedes García González, Elena, Ana Belén, and Cristina. And I want to thank all the funding entities because this is funded by the Ministry of Agriculture, Fishing, and Food. Thank you very much.
Francisco, I'd like to congratulate you for the dissemination of your project because that has made us be more interested in microalgae. And to finish the papers of this morning, we're going to have Javier Velasco now from BioIberica. BioIberica is going to talk about prebiotics and probiotics in agriculture and why not both. Good morning, everybody. It's a pleasure for me to be in such a gathering with lots of interested people in the use of microorganisms for plant health. And it's a pleasure because in my professional career in the last of 25 years, 30 if I take into account my PhD, I tried to make the most of microorganisms, make them produce, make the antibiotics, functional ingredients, prebiotics, probiotics, and at some point even biofuels. So I'm very, very happy and satisfied to see that in the plant health sector, microbiome and microorganisms are so important. When I say that I've been doing this for 25, 30 years, I can tell that I'm getting old in general terms. And getting old has very few advantages. We could say that there are two advantages. One is that the alternative is even worse, that is dying. And the second one is that if you are okay, you can learn from your mistakes and you can learn from your previous experiences because otherwise you're older and you're still dumb. So it's better to be older and wiser, learning from your mistakes. And I'm going to tell you something related with a mistake that I made in one of my previous experiences that I think can be relevant and can help you understand the context where we find ourselves now. 20 years ago, I was working in a company that was called Puleva Biotech. Now it's called differently and has been acquired by a big group. In this group, Puleva Biotech, I had there were lots of people that knew a lot about animal nutrition, great experts that there still are great experts in human nutrition, in food. Children food is like the Formula One of nutrition. Most of the ingredients that are going to be applied in other sectors for human nutrition for adults are first developed for children nutrition. This includes probiotics, prebiotics, omega-3, nucleotides, calcium, vitamins, etc. All of this is being used in children nutrition. Was used in much in children nutrition at that point. And we also knew what the bifidus factor was. It's a component of maternal milk It was discovered a bit before that that it was an oligosaccharide, prebiotic in maternal milk, and that means that children that are breastfed, have more healthy gut, gut microbiota. In Poleva Biotech, the team of researchers we discovered something that was very important. We managed to isolate bacteria in a human healthy maternal milk that was non-polluted. This is the first time that was discovered. Up until that time, we thought that maternal milk was sterile, and there are some bacteria, some, and we, not myself or my department, but it was discovered that there was an action mechanism that for bacteria to go from the stomach, from the intestine to the maternal milk, and with this prebiotic, the gut flora was developed in kids. So, being a company, we decided to produce this bacteria, to develop it and incorporate it in ingredients. We always think that in children nutrition there is a gold standard, that is that optimal nutrition is the best thing is always breastfeeding your child. All the new formula is getting closer to this standard, but we always say that the best option is to be breastfed. So we started working on this discovery, that we patented these strains, and we got confident, and we proposed to have a plan to produce these probiotics, because we're going to be the kings, we're going to get very far away, we're going to produce tons and tons of bacteria, we're going to sell this everywhere, because this is incredible, this is amazing. Okay, the finding was interesting, the product was good, but many years have gone by until those probiotics and those bacteria became commercial products. I don't know what happened with the plant, because I lost kind of track, but the plant took many years to have production problems. It was always at 10%, working at 10%. And was the product bad? No, it was good. The finding was important. It was very important. What was the mistake then? And what was my mistake there? Selling expectations that were too high. And I think that this is what I want to convey today, that having a look at the evolution of the sector of probiotics in humans and in animals, now in plant health, we've reached a time of a hype moment in the use of probiotics and bacteria in plant health. And this is okay. This promotes the creation of new companies, new businesses, but it's dangerous. The generation of expectations that are way too high. And when you go too high, well, the slope is big. So we don't need to try to sell that bacteria or fungi are the solution to all the problems on the field, or that this is the panacea. I would like to avoid terms like change of paradigm, breakthrough, a revolution like we heard yesterday. We're on the right track, but we have to keep calm. So in this sense, I would like to express what we're doing in BioIberica in this field. The starting point in BioIberica is products that are mainly prebiotics. And those of you who know the company know that our main source is L amino acids, obtained of animal origin, and we've proved. And yesterday, Alberto Acedo from Biomakers showed a paper where we proved the effect on the increase of biodiversity in the soil with the use of our prebiotics, these amino acids and peptides. And we start from that with a product that is over 30 years old, that is still working, and we wanted to take a step forward. As I was saying, we wanted to go from the adequate nutrition to optimal nutrition, not to reach the maximum. We're going to take a step forward. And with this, following this philosophy, we've been working on the incorporation of living microorganisms to our products. And what I'm going to present is how we can do this. Well, we knew that the strategy had to be to find, it was also explained yesterday, a microorganism that was a survivor in all senses, that could survive in our product, in the soil, that was capable to colonize it, that was capable to have the tools to attack and defend, to remain in a complicated environment as it is the root. And we started working with different options, different bacteria, and we selected, and I'll tell you more about it, bacteria, and we wanted to make it different and make a difference. How can we this bacteria that is naturally resistant and can naturally generate weapons to attack and defend, how can we make it a bit better? Our starting point were some compounds of ours. These extracts rich in L-amino acids, and they promote growth of some bacteria in the soil. Okay, let's cultivate it with this same raw material and see what happens. Let's see if this helps to have these tools to defend itself better. And at the end, we're going to combine bacteria, metabolites produced by the bacteria, and the prebiotics that are already included in our extracts, and we generate a product that exists. We've patented this product, and um, it's a prime intake, the strategy prime intake. Where does this microorganism come from? um, in the following the line of what I was saying before, not creating false expectations. If someone says, "Is your mechanism the best one in the world?" Well, probably not, because we haven't tried all of them. Can we compare ourselves with others mm, that we have access to? Yes, and we're doing well. In some areas, we're doing better. In some others, we're doing worse. Is it the best way in the world? No, it's very difficult to compare with all the microorganisms. Yesterday, it was said many of the microorganisms in the ground, in the soil, sorry, some of them are not identified, and some of them cannot be cultivated. So if you say that is the best one, it's kind of pretentious. So where is this microorganism? It's in the mouth of the Velez River. Bacillus velezensis is called like this because it was found in the sediments of the mouth of the river Velez. And this is a special environment. You need these weapons to attack and defend. There's a great concentration of salt, with spills, with nitrates, and the fact that it comes from there, it's relevant. There can be other pathogens that are useless, but it's important anyway. Side comment, because taxonomy, I'm going back, sorry. The taxonomic difference between the species Bacillus velezensis and Bacillus siemensis was made in 2017, so it's quite recent. And in fact, it's something that we want to, when we want to teach some taxonomy to the ministry, because maybe they think that the taxonomic classification is like the same as in mammals, and it's quite different. I'm going to skip the in vivo trials. Well, I'm going to talk about now the in vitro trials, fixation of nitrogen, siderophores. But the most important thing that I'd like to highlight is why or what the fact that this bacteria can grow and be cultivated in this base of our own raw material. What makes it important? Something that we managed to verify later is that the fact that we could cultivate this bacteria in a crop with our protein, it generates some metabolites in a higher amount. Here you can see amino acids, precursors of auxins, gibberellins, and vitamin B. This is all positive. We are finishing validating this, but the fact that the bacteria grows in its own environment, it means that when we add this bacteria in a series where the same prebiotic is present, the bacteria is going to grow better because it's got the tools, the enzymatic tools, to be able to colonize the soil better when it has the combination of the prebiotic and probiotic. We've also seen the viability in the soil at enzymatic level. We can highlight the increase um, of the beta glucosidase activity. We're still working on this. 2 minutes, Javier. Now I'm going to show data of three trials: lettuce, peach, and pepper. Where we saw firstly, if we compare it with the control sample, that in the case of lettuce, the there is a considerable increase of diameter and of the yield and productivity with our product. In the case of peach, we see an increase in the number of fruits and in the number of Brix degrees. And if we compare it with other biofertilizers, we can see in the case of pepper, we've seen an increase in precociousness. Is it significant? Well, it's not spectacular, but it's significant. But we see that we are high, a little bit higher than other products. In this same trial with pepper, we've seen the increase in the capture of metals. In the leaves, it's very relevant, the increase of phosphates and in sulfur. And in the case of copper, it's quite relevant as well. If we compare it with our product, the root product that we've had for a long time, and with the control samples. And to finish off, I'd like to say that we have developed a new product. It's a combination of our root TerraSorb, which from that we've had for a long time, with L amino acids, and we've included a probiotic with its own metabolites that are produced, and it contributes, this increase contributes with nitrogen fixation, solubilization of phosphorus, and the creation of hormones, the production of hormones. Thank you very much, and we'll keep on working. Thank you. Now we're going to have a coffee break. We're going to take a little rest and assimilate all this knowledge that we've learned. Thank you very much.
Good morning. Let's start the last part of this session. Can you please get into the room and take a seat? We're going to start the last part of this amazing conference on microorganisms. We're going to welcome Luis Gonzaga Garcia from the Technical University of Madrid and Corteva AgriScience, who's going to be talking about the use of biofertilizers, indicators of sustainability, biodiversity, and public participation aligned with the new CAP strategies 2023-2027. Welcome, Luis. Thank you very much for being here. Good morning. I'm going to present a group of 18 researchers from 3 universities of the company Corteva AgriScience and from a center of the Ecological Transition Ministry known as El Serranillo, who have created a project proposal that is being assessed at the national plan. The first thing I want to mention about this project is that the company Corteva AgriScience has been acting or collaborating since 2019 with 3 businesses in a public and private collaboration. The first one was the University of Seville. The second one was at the University of Lleida regarding weeds. And the third one is a collaboration with the Technical University of Madrid on sustainability of integrated plant production, both in crops and in forestry. Regarding all these activities, since 2020, Corteva AgriScience has been participating in a change of agricultural paradigm for the management of the rhizosphere that has to do with the different editions of microbiome. From Corteva, they are articulating their part on bioefficiency in crops with 5 products that can be named as biofertilizers and biostimulants. 2 of them focused on stabilization and the efficiency of nitrogen-based fertilizers on the soil. Another one is a biostimulant related to atmosphere nitrogen. A fourth one on the nutritional capacity of seeds, and finally a biofertilizer. This idea of bioefficiency and the development of these products has to do with a collaboration with public Spanish and European research centers in the framework of H2020 project called Novaterra that assess the impact of pesticides in olive trees and in vineyards in the Mediterranean area. Many different papers have been drafted and prepared, but the really interesting part for us is the part of biostimulants and biofertilizers in different experiments. Since 2021, we started collaborating Corteva Agri-Science and the Technical University of Madrid to change the paradigm on bioefficiency, combining the idea of bioefficiency with a model we've been working on for years that's called risoculture. Both models, bioefficiency and risoculture, try to look to intensify agricultural production, not just sustainability of agricultural crops and forestry. Talking about our models, what we want to do is observing ecological patterns. We look for techniques that enable us to intensify the abundance, the mass, and the activity of roots, of crops, of trees, and symbiotic organisms that have to do with the rhizosphere in ecosystems and in natural forests. I don't know if I can point it out. On the left, you can see conventional agriculture, whether it's fruit trees or crops, where the abundance of irrigation and fertilizers uh, lead to plants not having long roots. While in the right, that's what we try to intensify. You know how to cultivate the air part of the plants, but we don't know how to intensify the root part. We want to intensify these symbiotic relations, reducing the use of chemical products, phytochemical products. That's our goal, and that's why we suggested this project that's called Plan Gestión, planes de gestión to intensify the sustainability of agricultural and forest biodiversity. And we named the project AgroFnet. The general goals of our project are, first of all, to improve bioefficiency of crops. Second, to develop indicators to measure efficacy of risoculture and fertilizers. In the last 30 years, if we analyze scientific papers on environmental indicators related to agriculture, we see that most of them deal with topics such as the impact of trees around plots or biodiversity associated to agriculture, but there's nothing about biofertilizers or the positive or negative impact they have. And the third aim is to incorporate the public participation, thinking that it generates knowledge, what experts can contribute with, so that we can generate models to help us making decisions aligned with the new CAP policies for the next 5 years. What are the specific goals? The first one, analysis of the impact of these biostimulants and biofertilizers on fertility, carbon sequestration, development of sustainability indicators, management of a decision-making model for farmers and for managers. I've got 8 minutes left. Okay. And then the incorporation of the use of biostimulants in different contexts, and the incorporation of opinions of farmers, and finally the sustainable use of biofertilizers and biostimulants, and the impact they may have on the new strategies to be implemented by the CAP the green agriculture, adapting to climate change, biodiversity preservation, and social diversity. This has been articulated into some tasks. A first stage regarding sustainability application, selecting indicators, working with them, analyzing the impact on the use of biofertilizers to stimulate the soil activity, generating algorithms from all this data and from um, surveys to farmers, validation with public participation of the results from the models, and a decision-making model that's useful and that can be applied. Regarding microbiome, what does this project have to do with the soil? We incorporate a hypothesis on bioefficiency. That's the idea of 2 plus 2 equals 4, or does it equal 7? That means integrating different biofertilizer and biostimulants can lead to a higher response. So 2 plus 2 can make 7, can equal 7. And then hypothesis on rhizoculture. It's how we are getting closer to the model that I try to show. In that sense, both human beings and many organisms have had the skills to grow our niche. And that challenges natural selection laws, because at the moment when you change the environment regulations, the situation changes. So this niche construction is the process whereby organisms actively modify their own and each other's environment. We knew about it for different animals, but we didn't know about it for truffles, for example. They generate this situation you see on the screen, and for 30 and 40 years they eliminate everything except for the host tree, and that drew our attention, that called our attention, and we got to the idea that there are fungi that are farmers. So truffles, they increase the amount of substances they release, they block certain substances, and the burning effect appears, and this is like a feedback model. A niche is being built. We explored that outside the Mediterranean. We went to Oregon forests with other fungi with different soil conditions. Again, truffle in Oregon does the same thing. It changes the soil pH, it blocks nutrients. And here what I want to show you is that this is like an agricultural landscape. But in this case, farmers are the fungi. We have analyzed how they do that, the different tools that these microorganisms use to do so. We see on the graph the amount of mycelium and the amount of calcium, and calcium blocks magnesium, phosphorus, etc. So in the end, there's a modification in the soil for the fungus to control the trees. This is something that has been published. Is if we take seeds and do the same thing with them, that is increasing calcium and pH on the soil, growth patterns change in the roots as they smell calcium, they change the way they grow, and that's an endogenous response from the plant. Who knows that it's going to have to ally with the plant. Okay, 2 minutes left. The results suggest that there are mechanisms that generate these areas generated by fungi. And all around the world, we're collaborating with other universities to see that this happens all around the world, this calcium accumulation. So all of those strategies can be applied in agriculture, and we can get to systems that with biofertilizers also in forests we get to creating tools that can lead to the change of paradigm. And that's all. Thank you very much.
Now we're going to welcome Camino Garcia from AEFA with her papers, Current Situations and Perspectives of the Regulatory Framework of Biostimulant Products. Welcome, Camino. Good morning, everybody. I'd like to thank the organizers for inviting us once again. I represent AEFA, the Spanish Association of Manufacturers of Nutrients. Many of the companies here that sponsor and participate are members of this association. Before I start, I'd like to say that my paper is like the most boring one, if I may. Mine is more related to legislation, so it's kind of a little bit more boring. But the good thing is that I haven't got much time, and I'm not going to get into detail in any technical concepts. But for those of you that are not familiar with legislation of this kind of product, well, I just want to give you like an overview and give some context to show the work behind the putting biostimulant products in the market. I'd like to give you some information. As you know, biostimulants today are are very important now. The the European industry of biostimulants is a leader at worldwide. The companies in AEFA collected data from last year, and the estimated value of sales is of 250 million euros in 2022. The farm to fork strategy, the growth of ecological products, well, biostimulants is one of the solutions for this to achieve all these aims imposed by the European Union. But there is a large demand of this kind of products. It's been proved that they're useful. However, until last year, July 2022, there was no regulation or not even a definition at European level of the concept of biostimulant. What did this generate? This created chaos, enormous chaos in the sector that generated situations of mistrust for consumers and farmers, and complications for the industry to introduce it in the different markets, to comply with the different requirements of the different countries. and also competition problems because each one had to interpret how to introduce those products in the market. So even if um, it seems to be against what we do, against this freedom, if I may. What we've been asking from the sector for a while is a regulatory framework that is adapted and clear legislation about biostimulant products. So we move on to the regulatory framework in Europe for biostimulant products. We are always talking about biostimulant products. I'm not talking about biocontrol, phytosanitary. No, those things follow different regulations with different requirements. We're only talking about biostimulant products that are included in biofertilizers. So the regulation of stimulants applies. So there are two regulations, two things that you have to do to market them according to the European regulation or according to the national regulation of each member state. Then you have mutual recognition through the regulation EU 2019-515 with different requirements that we're not going to talk about now. So to these two regulations coexist, and the manufacturer has to decide how to introduce the product in the market. We're going to start with European regulation. We worked for a long time on this regulation. The previous one was dated 2003, and it only regulated inorganic fertilizers, inhibitors, and some amendments. In 2019, it was applied. It was approved, and it was implemented in July 2022. There is this optional harmonization. Both regulations can coexist, the national one and the European one. And it has a different and new approach of what was regulation of fertilizers before. It's a much more open regulation that suggests functions for products and categories of components with different combinations of these functions and components. If you meet all the requirements, you could have a fertilizing product. The objectives. This regulation was part of the action plan of the European Commission regarding circular economy, and one of the main aims is to promote innovation and development and the introduction in the market of new products, contributing safety, efficiency, quality in a free market within the European Union. So to introduce in the market a fertilizing product, in Annex 1 of the regulation, there is the function, you can find the functional categories of the product, the functions that that fertilizing product needs to have. There are six groups, plus the category of function number 7, which is a combination of the previous ones. That goes from CFP1, only manure fertilizers. Number 2, liming amendment. Number 3, soil improver. Number 4, culture substrate. Number 5, inhibitor. Number 6, biostimulant of plants. We also have in Annex 2, we have defined different materials of category component material categories for fertilizers, that today we have 15 CMC that are perfectly defined in Annex 2 and described, and to use any component it has to be included in one of these categories. If the product does not meet the requirements of any of the categories, it would not be able to be used in the formulation of fertilizers with the CE marking. And it has to meet some labeling requirements explained in Annex 3. Once you do all of this, it has to you have to subject it to the conformity assessment, where it says that it's a certification saying that you meet all the requirements. So for the first time, one of the most important topics here, for all of you here, is it's the first time that a definition is given of biostimulant of plants, and it's the one you have on the slide. I think it's been repeated in the last few days and years. In EU fertilizer, the function of which is to simulate the processes of nutrition in plants, regardless of the content of nutrients of the product, in order to improve one or several of the following traits in the plants and its rhizosphere: efficiency in the use of nutrients, tolerance to abiotic stress, always abiotic stress. We never talk about biotic stress. Characteristics of quality traits of the plant, and mobilization of nutrients in the soil. This regulation has subtypes: and microbial origin, amino acids, proteins, polymers, plant extracts, and microbial origin, non or microbial origin, and microbial origin that have to be microorganisms included in the CMC7 defined in the regulation, that today are only these four: Azotobacter, mycorrhizic fungi, Rhizobium, and Azospirillum. Apart from this, the rationale of these effects of biostimulants has to be proved with effectiveness trials with different parameters, and the different types of trials are defined in the technical specifications or harmonized standards that are yet to be published, that will go hand in hand with the regulation. What we say, there are functions that these products have to have. We also have the components that have to be in the product, and then you have to follow a conformity assessment procedure. What is this conformity assessment procedure? It's a process carried out by the manufacturer with the aim to prove that the product meets all the requirements of the regulation. The regulation sets 4 modules depending on the risk that this product may have for human health or for the environment. So it's more demanding or more strict to least strict. Module A is just self-assessment. The manufacturer can carry out or prepare this technical documentation and justify that that the product meets everything, until module DY, that is, and the B must C or the D1 are more strict, that imply the accreditation, the certification of a body that has to be accredited by the country where you request the certification, and they're going to express conformity to and say that the product meets all the requirements. This would be the way the European regulation works. It's very complicated and has some problems attached to it when interpreting it. At national level, well, at national level, we have legislation on fertilizers, Royal Decree 506/2013, and we have to say that we're lucky because it includes diversity of products, and that can also include biostimulants. As I was saying, Royal Decree 506/2013 and subsequent amendments, and its structure is more similar to the former European regulation of fertilizers that we have, with specific types of products. We have 7 groups of products: inorganic compost, organic compost, organomineral compost, other compost and special products, liming amendments, organic amendments, and there are other groups refined as to content of nutrients, obligations, limits, and according to which one of these categories can be included with all, then you can market it. Fertilizers that have organic components, groups 2, 3, and 6, need to be registered at the National Registry of Fertilizers, and the same occurs with 4.4, products based on microorganisms. You have an example here. For all the groups of fertilizers, you specify all the requirements, and if your fertilizer meets the requirements, each and every one of them, you could market it this way. In national legislation, there's a problem. The term biostimulant does not appear anywhere. However, there is a group of products, group 4, other compost and special groups that have these traits apart from the nutritional contribution to the plant. Group 4.1, special groups with amino acids, humic acids, silicon, extract of algae, mm, different diversity of groups that contribute nutrients to the plant. Then we have, since 2016, special products based on microorganisms that include mycorrhizas, non-mycorrhizic organisms, and this group was included in 2016 through Royal Decree 999/2017 and allowed in a very innovative way to include microorganisms in national legislation regarding fertilizers. To put these microorganisms in the market, you cannot only research and prove that they're useful and effective. You have to register them. It's a very demanding process. You have to meet the rest of requirements of the rest of products to register them in the registry. You have to present a collection, protocol of isolation, growth, and you need to have a positive report on cultivation trials. I think yesterday there was a presentation on these effectiveness trials, and this has to be done by an external, a third-party body that certifies that you abide by the requirements. This is something quite basic as to how legislation regarding biostimulants works. What I'm going to show now is that in spite of the fact that national and European industries are very powerful and there are many alternatives and many effects are being seen, all of these products at the end of the day have to follow legislation, otherwise they cannot be marketed. So here we have an important problem, we have an important barrier, because the pace of research is much faster than the pace of legislation of the public administrations. So it's a complex situation because a lot of the things are developed and then there's like a halt because it is not included in legislation, so you cannot market it. Some of the concerns, to mention a couple, in the sector right now with the new European regulation, well, microorganisms. We've been seeing for 2 days the effects of microorganisms that are being researched. However, in the European regulation, there are only 4 that are allowed. So this list is undermined, and the possibilities for the industry are undermined. There are procedures to update this list, but these are long procedures that involve at least 2 years until you can expand the list. These are byproducts of our animal origin. We still don't have byproducts of animal origin in CMC 10. So these products made from hydrolyzed proteins from of animal origin amino acids have not been marketed yet because they're not included in the regulation. Then Rich Plus, there's a lack of harmonized standards that have not been published, and now we have to work with technical specifications that may be subject to changes. Legislative deadlines or working timeline. There's a lot of pressure, a lot of industries required from the industry. Differences between the criteria, the assessment criteria of notified bodies. Depending on the notified body you request a certification from, they demand more or less. Depending, if you want to have a European harmonization, then this shouldn't be like this. And there are many concerns that have the industry kind of worried because the development of the products depends on them. And to finish off, well, we are AEFA, the Spanish Association of Agronutrients Fabric Manufacturers, made up of 59 companies based in Spain. And the idea is to be the voice of this sector and to be the communication channel between the industry and public administrations, organizations, other associations, to try to help the sector move forward and convey the real needs that the sector has, the field has, and that to try and be consistent within regulations and legislation. You have here the companies. You can visit their websites. What do we contribute to the sector? We participate in all regulatory and legislative fora. We participate in specific work groups. We disseminate the development and innovation of our companies. We highlight the work carried out by this industry, that all this research involves efforts, research efforts by the industry or public administrations, and we need to highlight this. We promote different initiatives within the sector. We contact different associations and we try to exercise pressure for this regulation to move forward. To sum up, we promote the technology transfer, research and development. And to finish off, I'd like to say that biostimulants and microorganisms are, it's clear that it's a tool that we need to achieve all the aims set by the European Union. It's key that the regulatory framework is adapted to the needs that the sector has. And we have to all exercise pressure. Yesterday, one of the speakers mentioned this, that we need to exercise pressure so that we move forward, the industry moves forward. Spain is leader and pioneer in biostimulant products, so we need to emphasize this and convey it correctly and talk about this great effort. So thank you very much for your attention and for helping the sector. You have our contact details there and you can contact us. Thank you, Camino. Thank you for sticking to your time.
Now we're going to welcome Javier Pozueta from Timac Agro, who's going to be talking about innovating research lines, volatile biostimulants of microbial origin. Welcome, Javier. Good morning. My name is Javier Pozueta. I'm a research professor at CSIC, and I work in Malaga at a mixed institute from CSIC and the University of Malaga. First of all, I would like to thank the organizers for inviting me to give this talk. I'm really honored, not just for the fact of having the opportunity of explaining results and 15 years of research, but because they've given me the opportunity of participating in a forum such as this one, where I feel really comfortable with, because we not only discuss research aspects, but where we transfer results to companies. I have collaborated with companies in the past, so I'm really satisfied with this opportunity. As I only have 15 minutes to explain this story, and due to the fact that I don't have too much time to thank, I want to show all the institutions that have participated in the project. The Institute of Agrobiotechnology, Niigata University, Japan, in the University at the Czech Republic, and the Institute for the Mediterranean and Subtropical Horticulture, where I work at. And this is the group. On the left, there's the group that I had the honor to lead for approximately 20 years in Navarra. Some of the members, Edurne is here at the audience. On the right, there's the group that I created in Malaga 3 years ago, where we started working with microbial-based biostimulants. I also would like to thank the institutions that have funded This research, and especially Team Agagro, we're working with for 3 years in order to look for microbial based um, biostimulants, not only to identify but also to characterize the action mechanism or the mode of action that enables us to differentiate between magic potion and something that really works, taking into account the hard regulations established by the EU. These started, uh, this is a serendipitous case. On the left, you can see a guy with a blue polo shirt. He was doing his PhD. Today he works at the University of Milano, but Nacho at that moment made a mistake that you can see in the pictures on the left. At that moment we were researching things related to the metabolism of Stark, and he did the characterization of plants which were polluted or contaminated. And we found really surprising results. The picture at the bottom not only shows Nacho, but also two other colleagues that are sitting here today who led a project to promote taking this technology to the market. What we found is that this contamination that we had on a cultivation dish led to an overaccumulation of starch, but with time we found out when we exposed plants to fungi or microorganisms in a controlled way and with no physical contact, we saw that bacterial species and fungi species catalogued as beneficial or pathogenic, they all accelerated the flowering and increased the size of the plant. Just doing what you can see on the picture. Putting microorganisms next to the plants. Therefore, plants perceived substances, volatile substances emitted by microorganisms. And this let us think that microorganisms, before colonization, they established communication, because these fungi and these bacteria include species that normally do not interact with plants, at least in nature. But they boost growth and they accelerate the flowering process. So this was sending a very important message. That's stark. And let's see what happens. On the left, we have the mock, and those in the middle are the ones exposed to volatile compounds by Alternaria alternata, and on the right, Penicillium arantigriseum. But we can see the root system that it creates. It is spectacular. The production of root hairs is multiplied by 20. And our task is to look for the biomolecular bases that regulate these responses. Associated with these changes in the root architecture, we saw that there was a higher photosynthetic activity and an increase on the content of soluble sugars. That's the primary metabolism that's activated. That's connected to the secondary metabolism. On the top, we have glyceraldehyde phosphate. This is a really complex family with many hormones, with all of the elements you can see on the screen in green. So we have a target for these volatile compounds. That's the alteration of something that's as delicate as the sopranoid metabolism. This is a very complex figure that shows that plants standing next to microorganism compounds react to and cytokinins that regulate the photosynthesis, cell division, etc. From the huge amount we accumulated with different omic techniques that shouldn't impress us because we are familiar with them. Now we are at a different stage. We're working with artificial intelligence, etc. All these models have been done with natural intelligence. Of course, they are associated with a huge amount of effort, but they generate a high satisfaction. The first data we gathered from transcriptomics showed a change in the plant transcriptome. We saw that there were packages associated to the different pathways, and we thought this is regulated from the transcriptomic point of view. But this didn't last long because we soon realized that all the treatment, these are changes that we observed in a plant, but everything was subject to volatile flora alternaria alternata. The same thing happened to plants exposed to high UV light rays, etc. This is what happens in the plant proteome. Changes that happen that do not match changes that we've seen at the transcriptome. Just 10% of proteins that are expressed are codified by expressed genes. So we have to be careful when studying biostimulants in plants. We cannot base the research on transcription, just on transcription. And this means that apart from changes in the proteome, there are other changes after translation. So these are years and years of research. These 3 slides show years and years of research. And they show the mode of action of these compounds. And this is at the root level. And it's also very complex. But basically, it is saying that changes that happen at the root level and in the plant are communicated. This is what we are working at the moment. What are those communication mechanisms between both parts of the plant? These are the conclusions. One of them is that bad guys can be good guys. And this was very important because at that moment we thought that only good guys could generate positive responses for human beings and for the plants. But that wasn't the case. Whenever we talk about pre-colonization, then we wanted to take that to the field and we wanted to treat plants with extracts, filtered extracts obtained from fungal culture And we were asking the blue and red dots represent microorganisms, positive and negative microorganisms. And we were wondering, does soil application of cell-free filtrates of beneficial and pathogenic fungal cultures improve yield crops of agronomic interest? And the answer is yes. Plants treated with cell-free filtrates increase not just the yields of pepper plants in greenhouse culture under greenhouse conditions. 2 minutes, Javier. Thank you. And then we ask about ourselves about volatiles, if those mixtures, if those volatile compounds can play an important role in this response. And the answer is yes. What we did here was to distillate all these compounds, chlorine, turn into something transparent, and we had the same response, an increase of production. And again, we ask ourselves, does it have an effect on microbiota in the soil? So we analyzed it and we saw that there were changes. We used filter substances and distillate substances in irrigation water, and we saw changes. And there were some species, such as the ones you can see on the screen, that were beneficial. Microbiota responded the same way in all the cases. And the same happened with fungi. So the conclusion is that those compounds, volatile compounds, that we have in these filtrates with fungi and bacteria are essential in the plant response and the microbiota response to the treatment we're applying. So within those thousands of substances, there are a few volatile compounds that are responsible for all this response. These are the conclusions, and these are two papers where we work with this topic. Javier. Okay, and that's it. And that's all. I just wanted to show you some videos and the results that we had with water stress conditions that have been spectacular. Thank you very much for your attention.
Thank you very much, Javier. Very interesting. Even if you didn't have time to finish your presentation, we're going to give the floor to Alberto Vago from the Sidene Experimental Center that's going to talk about new developments in the use of mycorrhizae. Welcome, Alberto. Before I start, I'd like to thank the organizers for existing, basically, because you have to be very brave to organize something like this. And thank you for being so patient and having me here. Another edition. At the beginning, I'm always like in panic because I'm afraid that I'm not going to meet the expectations. Oh, this is the crazy guy about mycorrhizae again. But I wanted to say something following the line of the last speaker. Don't you think it's incredible? All of this is incredible. I know that we're here. I'm a researcher. Maybe you even find microorganisms disgusting, but don't you think that there are so many things here? The previous speaker said something. The bad guys are not that bad. This is yin and yang. We need to know that not all the bad weeds are that bad. Maybe they have mycorrhizae that help other plants. Anyway, I'm going to talk about we say new developments in the use of mycorrhizae in agriculture. As this presentation has a different approach, but I can't start without. I know that most of you are experts in mycorrhizae, but I'm a bit older because I've been working on mycorrhizae for 35 years. Some of them say 5, 20. Well, no, I'm older. 35 years working with mycorrhizae. I'm still passionate about them, and that's what I try to convey, my passion. So mycorrhizae are mutual symbiosis between most of land plants and fungi in the soil that are fungi forming mycorrhizae. You have the typical mycorrhizae types. We have, for instance, estomycorrhizae. It's the best way to see it is in truffles and that are associated mainly with forest species. The ones that we're going to focus on are the ones that have their niche in agricultural plants. Um, Arbuscular mycorrhizae are known as endomycorrhizae, but we should not get them confused. And the important thing is that I'm saying is this intertwinement of the fungus and the plant creates some symbiosis that is so intimate that they're not independent anymore. It's a supra-organism, something new. It's not roots, it's mycorrhizae. I can say that mycorrhizae. I'm talking as if it were my kid, my baby, my son or my kid is the most intelligent one. Well, mycorrhizae is the mother of all microorganisms in the soil. It's the one that hosts everybody and has an intertwinement with the root. I like this slide to show the distribution of plants that have arbuscular mycorrhizae in the world. So 100% is blue in areas that are warmer and Mediterranean. They are full of arbuscular mycorrhizae. If we go to ectomycorrhizae, we see more the boreal areas in forests. Then we have ericoid mycorrhizae that are more residual, and then we have non-mycorrhizic plants that if we compare them with arbuscular mycorrhizae, ours are less. So if we take now we're talking about agriculture, crops with arbuscular mycorrhizae is about 90% of the crop surface. So it's very important. Very quickly, I'm going to talk about the traits of arbuscular mycorrhizae symbiosis that are very old. They penetrate these root cells and they create these fantastic structures, arbuscules. So the root doesn't have rhizosphere, but mycorrhizosphere, and it's going to transfer nutrients to the plant, and they're invisible. But as you know, they are fundamental. And this slide is quite interesting. We see very illustrative. We have the two parts of the root. One part is not mycorrhized, and the other one it is. And the yellow triangles, we see the depletion area of, it says phosphorus, but it could refer to any nutrient in a plant that is not mycorrhized. On the right hand side, we have this area of phosphorus depletion and we see that the nutrients and water is going to be used there, and that's why arbuscular mycorrhizae is so powerful. Mycorrhizae is the mother of all microorganisms. When the fungus goes out, mycorrhizosphere is produced, and there are a series of interactions with microorganisms of the mycorrhizosphere that are intimate interactions. This could take for not one conference but 50 conferences. When I talk about this, when I always think, why don't I have 3 hours to discuss this? There's a bacteria. And I want to talk about the bacteria. I want to talk about trichoderma. I want to talk about everything. But anyway, this intertwinement that we have is what we have to bear in mind in mycorrhizae. Very quickly, benefits of applying mycorrhizae. Increases the surface of exploration and nutrients of water. Um, Absorption of nutrients that are less assimilable, like phosphorus, nitrogen, and other micronutrients. Finds a balance, increases survival and production of plants, increases the yield and resistance to pathogens. We already know this. Impact of mycorrhizae. Well, I'm going to skip it because haven't got much time. So mycorrhizae are fundamental, and why haven't we used them? Because we didn't know how to use them properly. This is a slide that I use a lot, and you're going to think, change this slide, please. That you always say the same thing. But the base of this is that the fungus that creates mycorrhizae is going to condition research and the development of inoculants. What happens that, as I have mentioned a lot of times in other talks, the form of cultivating mycorrhizae has to be with a plant, and this was the conventional way of cultivating mycorrhizae that is still used in some production of inoculants. There were lots of problems that you can read on the slide, and that made it almost impossible to understand the mechanism, the genetics of the mycorrhizae. But in the 60s, we started to see a movement, the same as in bacteria, that at the beginning nobody studied biochemistry of bacteria. We have to study the soil to see the relationship with the soil. But if you study it in a petri dish, well, we try to do it in an in vitro culture. And there are some milestones here in mycorrhizae. One is that in 1988, in the first time that this was presented at a conference, the big bosses of mycorrhizae said, this is pollution, this is not useful. And it took us 10 years. A group of crazy people met in Quebec and realized that it did work. And the fact is that since the year 2000, all labs in the world are using mycorrhizae, in vitro mycorrhizae. And okay, here comes a scientist to talk about his research. Why is this important? How can I bring this advantage to large scale production, and how can I apply it? What I want to insist is that I think sincerely that the future of any mycorrhizic inoculant goes through in vitro culture. But we may think this is very difficult, only a few can do it. Well, I'm going to tell you a story if I'm allowed. In 2000, when I came from Canada, I brought something and I found my colleague Victoria Cano, and she said, but you have everything in your hands. I was studying my biochemistry of mycorrhizae. Why don't we apply it in inoculants? And in 2005, we patented the first one. In 2005, it was patented, but the patent was over some years after. Sometimes you don't know that. You just have to prepare for yourself because there are many things that have to be done. In 2007, the two of us set up a business because we saw that inoculant had a future. So we set up a business to highlight the importance of this inoculant, and it was called Glomiger. In 2013 and 14, biology very good, but math is not very good. So I've got 10 minutes left. Is that it? Okay. Thank you. Thank you for your kind round of applause. Let me go back to my mycorrhizae where I feel comfortable. So I was saying that in Mycovitro, we had the exclusive license to explore this technology. and it was very difficult. It took a lot of effort. I'm very really excited to see that in this forum everybody uses the term mycorrhizae. They pronounce it properly. Nobody messes it up. No, everybody knows what a mycorrhiza is. Even in legislation, we can see the term mycorrhizae. And at the beginning, in 2007, we went up to the to administration in Madrid, and they said, "What is this? Is this a bacteria?" They have no clue. They had no clue, but now it's different. So it took a lot of effort because when we imagine in the two of us researchers, we went to see farmers with a product that was like a diamond that had to be improved. And so we went up to the farmers and said, "We have this product. Yeah, leave it there on the shelf and I'll try it at some point." Someone else said, "Microbiocide? I know, I don't want to even hear about that. I've been already fooled with this because they said it was inoculant." Somebody said that we have to be take into account the fear of false expectations. People were thinking because sometimes we think that it's the panacea, but one thing is to have the panacea and a different thing to have the fungus or some plates, and a different thing is that you take it to the field and it works. I have so many things to say. It's very important to have the adequate fungus, but if the formulation is not good, what do you want the fungus for? Or if you have good formulation and the fungus is not working very well, what do you want the inoculant for? So we need a balance. So here I urge you to talk with researchers, to talk with us. We have so many things to give you. I insist in 2025, this is going to be crazy land. Anyway, in 2007, we set up the business. We found it very difficult, but we were obstinated. So we were stubborn. So in 2013-14, we had a niche in the market and we called the attention of some companies, one in the US, and we signed an agreement, and it was marketed as Mycos Gel in North America, another one here in Spain that was marketed as Mycogel, the brand Mycogel that you all know. However, this is very important. I'm talking about a patent. The patent is like the diapers, the nappies of the baby, and you have to take care of the kid since the kid is born until the baby turns into an adult. And so having a patent is very important, is the base, but the most important thing is the technological key that we collected with all our experiences, all our trials, or the improvements we made starting from that base that belonged to that company, that made that the inoculant was more effective, had better yield, worked better and longer. In 2018, for different reasons, the company closed up and transferred the technological key to a different new company. The technological knowledge is immense, and we would like to put at the disposal, at society's disposal. I've got 5 minutes left. Okay, perfect. And that year we signed some trade agreements, but sometimes it is like a marriage. Sometimes it's successful, sometimes it's not. In this case, it was a not successful company. The company that markets microsize now, well, we don't have a very good relationship, but it's a pity, but we have to be positive, and bad things happen for a reason, and this has opened up our mind to make us know that we need to move, that we cannot remain like in a capsule. Microarchaea are too large, and we have to move in the world of microarchaea, because what we have now, even the gel, is a history now. We have lots of new things. There are lots of new findings that are there waiting for someone to push it forward. Why are we restricting to some specific strains? Something that's important. Everything I'm saying now, I would like it you to bear in mind that according to my knowledge, the best way to have inoculants that can be certified and validated and that are useful is using in vitro technology. Don't be scared by it because it's easier than you think. Why do we have to have one strain too? A mixture of strains, everything very restricted because those manufacturing manufacture one strain and make a lot of copies, but there are strains with a great potential and we can use them in crops. For instance, we talk about salinity problems, heavy metals. Glomus custos, they're in the river, in the Tinta River. There's a lot of pollution and it has more copper than a non-mycorrhized plant. But you need to know that it exists. Why do we have to stay with the presentation? There was a fight, and I'm very much aware of this, that powder inoculants, solid inoculants, gel inoculants, there are a lot of there have been a lot of presentations. For instance, a clue. What would you tell me if I told you that there's a way to super produce active spores, that the problem of spores, they stay dormant in the soil, but if they're very active in an amount that now you cannot imagine, well, that's something that it's there. There is a technological potential that it's enormous, and this is what I want to talk about. Or in the previous speaker said that the that without mycorrhizing the gel, it has some effects on the plant and the microbiota around it. But I'm going to go beyond. Is what I call the democratization of mycorrhizae. If you knew, there are a lot of stories about this. Marketing is like this. If you knew, if you knew the costs, the production costs that you have once everything has been said of an in vitro mycorrhizae compared to what you lose, you would have a laugh. We could reduce that price, that sale price. I'm saying this because recently a colleague from Guatemala that we've known each other for a long time said, "What a pity that costs are so high. 100 euros a hectare. This would be impossible to apply in Guatemala." So the farmer creates his own inoculum and can be better or worse. But if you reduce those costs, and I thought, yes, you can do it with this new technology that we can offer. So the takeaway message, no, let's not turn mycorrhizae into a lobby. These are the mothers of all the microorganisms. Agriculture needs them, so let's make them as accessible. I'm going to finish off with the characteristics of the good inoculant. This is like the mouse red book. And these characteristics are quite obvious. And to have a sufficient number of propagules, that they are viable and they are kept in the formulation you have. It has to be a good fungus and a good formulation, not to have other microorganisms that are annoying us, even though you can have microbial consortia. But we have to control them and not the inoculant control us. 13 seconds. This is like when a spaceship is about to take off. Not to have other materials that dilute the weight that we want. So it can be easily applied. We cannot go to the farmer to ask him or her to change everything to use the mycorrhizae. Mycorrhizae are more resistant than we could think, but we have to adapt them to each situation. The correct fungus for each situation, the correct inoculant for each situation, so that it doesn't have additives. Mycorrhizae, they said in 24 hours you can see the effects. Well, mycorrhizae has to get to the root and it takes at least 5 to 7 days. Don't tell me that this is magic. it has to be compatible with good practices, with good cultivation practices, and to have good results. I had another slide. mm, Talking about different fake news about microacai, but I've already presented them. But I don't want to oversaturate you, overwhelm you. So the only thing I want to say now is good luck. Support microacai, and thank you for your attention. And I'm at your disposal. Thank you, Alberto.
Now we're going to welcome Gorka Edice from Athens. He's going to talk about GPG bacteria-based biostimulants, improving the use of fertilizers as they increase the efficiency of the nitrogen use. Okay, just a little bit more. We're finishing. We're there. My name is Gorka Edice. I'm the Technical Global Director of Athens Natural Agrotechnologies, and I'm going to introduce I'm going to make this presentation named GPG bacteria-based biostimulants to improve the use of fertilizers, and we're going to go a little bit deeper. Athens is a company that's located in Tarragona. It's a biotechnological company that works in the reproduction of microorganisms. We're going to see mycorrhizae, trichoderma, and I'm going to show you how we do it with bacteria. In Athens, we have been working for more than 30 years, and it is a company that was born from R&D. And in fact, we are really proud of our R&D department. We have 2 international patents, one of them related to seed cover, seed coating, to add spores to the seed surface, and the other one relates to a vegetable fiber that can get water and for it to be bioavailable, not just for microorganisms but for plants. And in our NGA lab, it's Next Generation Agronomics, we're focused on omic sciences. In analyzing in a holistic way the action mechanisms of our products. Javier Pozueta explained that if we don't know what the action mechanism of microorganisms is, we don't know anything about them. We need to open up the soil and discover how it works to understand the best protocols and the application times. We have a library with more than 1,200 strains of microorganisms, and we have more than 200 scientific papers. Today we're going to see a microorganism that we isolated, that we promoted. We know its action mechanism, and we're going to see a paper. In Athens, we have certifications for environmental management, quality management. We have a warehouse with more than 8,000 square meters. And we can adapt to the market needs. But as you can see, the market is growing really fast, but we have space to go on growing with the market. As for the regulatory framework of our products, we have trichoderma. That's going to be regulated as pesticide at the EU, and for the US, or in less than a year, it's called Trichoderma atroviride 8010. It can be used for tomato, lettuce, for vineyards, for peppers, for cereal and corn. And we have other registrations that will come later on. As biostimulants, we mainly work with mycorrhiza. The regulation that Camino mentioned this morning that we're going to complete this year. And for bacteria, as we only include 4 species, as only 4 species are included, we use the Spanish regulation. Now we are expectant to know what the method to be able to increase this list is going to be. If you have any questions, you can ask Camino. On the production of mycorrhiza, we produce them in vivo. That means that we have host plants in tables, and therefore we control the nutrition. We inoculate the mycorrhiza at the beginning of the season, and we gather the substrate with the spores at the end of the season. Therefore, our fully automated greenhouses can respond to an inoculum that does not depend on external conditions. We're going to have a similar amount of spores in the raw material, and this standardization is one of the main worries of the microorganism industry, to have a consistent efficacy in the field. We have our own quality controls, but each batch is analyzed by the IRTA as an independent center. One of the advantages of using this system, this in vivo system, in greenhouses is that with the mycorrhiza, there are other bacteria that develop. We presented this paper in 2018. It is open access, you can read it for free. And here we identified 207 species of bacteria next to the mycorrhiza, and some of them had PGPR properties. Some of them promoted growth, others promoted the absorption of phosphorus, etc. These bacteria, apart from creating base for R&D, we took 2 of them to Bacilli megatherium, MHB M06 and 77, that combine 2 abilities: solubilization of phosphorus, and another aspect, the use of siderophores. We used it in the dosage